Today, we will examine the positive controls that the Nobel Laureates used in one figure panel of their published paper. A graph in the figure 3D, provided the critical data for developing the messenger RNA vaccines, and the work eventually led to the Nobel Prize in Physiology or Medicine 2023. The graph was so important that it was shown in the Nobel announcement video by the Nobel Committee too, and we called it the Milestone graph. After reviewing the positive controls, we will ask if we can use the term positive control throughout a paper. We will also discuss how a modified definition of positive controls will be helpful. Hi everyone, welcome to the Foundational Skills in Life Sciences podcast. I'm your host, Dr. Synaptologica. We discuss the skills that life science scholars learn, use, and improve throughout professional activities. Your benefit is expressed in my motto: learn the skills, enjoy academic life. Last episode. In the last episode, we reviewed the 4 positive controls in figures 1 and 2b of our Milestone Article 1. This is the paper published in 2005 by the laureates of the Nobel Prize in Physiology or Medicine 2023. The laureates were Dr. Katalin Karikó and Dr. Drew Weissman. I have added the links to the paper in the episode description. If you are a new listener of my episodes, please start listening from episode 7. It was the start of the mini-series of reading. Now, in figures 1 and 2b, one group of positive controls were synthetic compounds. They were poly-IC and R848. Poly-IC is a synthetic analog of double-stranded RNA, and it selectively activates the toll-like receptor 3, and R848 is a low molecular weight synthetic molecule, and it selectively activates the toll-like receptors 7 and 8. Comment 1. How to remember the actions of the 2 compounds. A quick comment in the middle of the last episode section. Just to let you know, we will mention poly-IC and R848 multiple times today. To make it easy for you in my episodes, I created a mnemonic for linking these 2 names with the toll-like receptor subtypes that they activate. It is made up of rhyming words. Poly-IC activates toll-like receptor 3, so poly-IC3, and R848 activates toll-like receptors 7 and 8, so R848-7-8. So the mnemonic is poly-IC3, R848-7-8. It is a little long, but I like mnemonics. I hope this mnemonic will help us read the paper. Let's get back to the last episode. Another group of positive controls were the unmodified forms of in vitro transcribed RNAs. For example, in figure 2b, they were RNA 1571 and RNA 730. Using all these positive controls as the guides for evaluations, the laureates found basically 2 results in figure 2b. The first result was that the unmodified in vitro transcribed RNAs activated all 3 subtypes of toll-like receptors, that is, toll-like receptors 3, 7, and 8, and the activated cells released inflammatory cytokines. The second result was important, but I forgot to mention it in the last episode because I was too much focused on the positive controls. Sorry about that. And the second result was that when the in vitro transcribed RNAs included modified nucleosides, the cytokine release was suppressed in comparison to the same RNAs with unmodified nucleosides. Different types of modifications suppressed the cytokine release to different degrees. This second result was especially important from a larger perspective. This is because if the cytokine release is suppressed when RNAs contained modified nucleosides, replacing the unmodified ones, then the messenger RNAs with modified nucleosides can be safely used as messenger RNA vaccines. They will have minimal or no inflammatory responses. They could lead to minimal side effects. But these experiments in figure 2b were done on the model cells, the human embryonic kidney HEK293 cells. So the natural next step would be to ask if the same results can be shown with natural immune cells. In other words, the natural next step would be to ask if the RNAs with modified nucleosides can suppress cytokine release also from the natural cells responsible for immune responses. Such results are shown in our milestone graph in Figure 3D. Today's episode. We are still at the reading step 11-6 about positive controls in a key figure panel. We will examine the examples of positive controls in Figure 3D, which contains our milestone graph. Experiments in Figure 3D. Here are 4 features about the experiments in Figure 3D. I will also point out 2 differences from Figure 2B that we talked about in the last episode. The first feature was that the laureates used the dendritic cells in Figure 3D instead of HEK293 model cells in Figure 2B. This was one of the 2 differences from Figure 2B, and this was the major difference. According to the YouTube video presentation by the Nobel Committee, dendritic cells are important in, quote, immune surveillance and vaccination, close quote. So the dendritic cells are at the forefront as the responders to incoming RNA. The second feature of the experiment was that the laureates prepared 2 different types of dendritic cells. Both of them were so-called primary dendritic cells obtained from the peripheral blood. One type was the primary myeloid dendritic cells, labeled as DC1, and the other type was the primary plasmacytoid dendritic cells, labeled as DC2. DC stands for dendritic cells. But these specific names are not so important to us outside the field. Rather, it is important for us to note that these are natural immune cells that would respond to the RNAs when the RNAs are introduced into the cells from the outside. The third feature of the experiment was that the laureates introduced RNAs or related synthetic compounds into these cells. And the fourth and the last feature was that the laureates measured the amounts of the released cytokines. 2 cytokines were measured in Figure 3D. One was the tumor necrosis factor alpha, abbreviated as TNF-alpha, and the other was the interferon alpha, abbreviated as IFN-alpha. And the laureates compared the released amounts induced by different RNAs and related compounds. These cytokines were the second of the 2 differences from Figure 2B. So, in Figure 2B, the cytokines were interleukin-8, whereas in Figure 3D, they were tumor necrosis factor alpha and interferon alpha. But these specific names of cytokines will not be so important to us outside the field. It is important for us to note that these cytokines are released from the immune cells in response to RNAs, and that if the cells release less cytokines, that would be a good finding. So, in summary, the laureates tested the effects of multiple types of RNAs and related compounds on the release of 2 types of cytokines from 2 types of primary dendritic cells. If we jump to the conclusion of Figure 3D, we can happily say that the message remained the same, even when the laureates examined different types of primary dendritic cells and different types of cytokines. The release of all tested cytokines from all the tested cells was suppressed when the unmodified nucleosides in the RNAs were replaced with the modified nucleosides. How Figure 3D looks. Here, I would like to describe Figure 3D a little bit so that you can visualize it easily. Figure 3D looks like a table. It will look like Figure 2B that we talked about in the last episode. I will tell you what are similar and what are different between the 2 figures. Figure 3D has rows and columns, like Figure 2B. The rows represent experimental categories, like in Figure 2B. There are 12 rows. Each row has a vertical axis label or category name, which indicates what RNA or related compound was introduced into the cells. and the columns represent the types of primary dendritic cells that were treated with RNAs or related compounds. These columns are different from those in Figure 2b. In Figure 3d, there are 3 columns or figure subpanels. The first column corresponds to the primary myeloid dendritic cells, labeled DC1, and the other 2 columns correspond to the primary plasmacytoid dendritic cells, labeled DC2. At the intersection of a row and a column, there's one table cell, and the table cell shows a horizontal bar, and the bar length represents the amount of the released cytokines under the specific condition for the table cell. These features were the same as in Figure 2b. But in Figure 3d, the measured cytokines were different. In the first 2 columns, it was tumor necrosis factor alpha, and in the last column, it was interferon alpha. Among the 3 columns, or figure subpanels, the first one on the left is the Milestone graph. So the Milestone graph was based on using the primary dendritic cells DC1 and measuring the released tumor necrosis factor alpha. Now that we have understood the basic features of the experiment in Figure 3d, let's talk about the positive controls in the figure. Positive controls in Figure 3d. In Figure 3d, 4 positive controls were used. 3 of them were the same as in Figure 2b. They were poly-IC, R848, and the unmodified RNA 1571. A 4th positive control was new. It was a synthetic compound called poly-U. The results with these 4 positive controls were interesting. Let's examine them. Positive controls in Figure 3d. 2 synthetic chemicals. First, let's talk about the synthetic chemicals poly-IC and R848. As we discussed in the last episode, these 2 synthetic chemicals were identified in Figure 2b as positive controls for stimulating specific subtypes of toll-like receptors when they were expressed in HEK293 cells. Poly-IC activated toll-like receptor 3, whereas R848 activated toll-like receptors 7 and 8. Remember, the mnemonic is poly-IC3, R848, 7, 8. In Figure 3d, R848 showed the longest bars in each of the 3 columns. It means that when R848 was introduced into primary dendritic cells, the cells released large amounts of cytokines, maybe through activating toll-like receptors 7 and or 8. This result was good. But poly-IC showed only very short bars in each of the 3 columns. It means that poly-IC did not stimulate the cytokine release effectively. So, most likely, the cells did not express toll-like receptor 3 to a large extent. Together, these results with 2 positive controls indicate that the primary dendritic cells expressed either or both toll-like receptors 7 and 8, but not toll-like receptor 3. I have one comment here. Comment 2. Statistics. I have described different bar lengths as long and short and compared them qualitatively. I know that this comparison does not sound conclusive to you. If we are to do a peer review of this paper now in 2024, we would most likely point out that the results in the paper were presented without any statistical analyses. In current papers, when we compare 2 or more measured values, we will need to include statistical analysis. We may conclude that the measured values are different only when the difference between the measured values is reliable and meaningful, as determined by statistics. Then we will say that the difference is statistically significant. This is the standard approach now. It was not the case with the Milestone Article 1. But I would say that there will be no problem for you and me when we read this paper. The Milestone article was published more than 20 years ago, before such quantitative analyses were introduced rigorously into scientific publications. And also, the main results in this paper are so obvious that a statistical analysis would have only supported the laureates' conclusions. Here, I just wanted you to keep in mind that in most papers now, we will need statistical analysis to say something convincingly. Back to the main content. Positive controls in Figure 3D, unmodified RNA 1571. The third positive control in Figure 3D was RNA 1571. It was one of the unmodified in vitro transcribed RNAs used in the paper. Again, we discussed it in the last episode, and we learned that in Figure 2b, the unmodified RNA 1571 was a positive control for stimulating the toll-like receptors 3, 7, and 8 in HEK293 cells. In Figure 3d, the unmodified RNA 1571 showed one of the longest bars, similarly as a positive control R848. This result means that the unmodified RNA 1571 stimulated the cytokine release strongly, and that it can serve as a positive control not only in the HEK293 model cells, but also the natural immune cells, that is, the primary dendritic cells. This was good news, because if modified RNA can be shown to suppress the stimulating effect of unmodified RNA on releasing cytokines, then there's a chance that messenger RNAs with modified nucleosides can be safely used as messenger RNA vaccines with minimal or no inflammatory responses. In fact, we can read such results in Figure 3D. The bars were long for the positive control, the unmodified RNA 1571. In contrast, the bars were very short and almost invisible for the modified forms of the same RNA. Particular examples were evident when the unmodified nucleoside uridine was replaced with modified nucleosides, pseudouridine, 5-methyluridine, or 2-thiouridine. A short note: the names of these modified nucleosides are not so important to us outside the field when we train our reading skills. Instead, it is meaningful for us to know that the unmodified RNA 1571 served as a beautiful positive control in Figure 3D, and that the modified RNAs suppressed the cytokine release. Positive control in Figure 3D, poly-U. A fourth positive control in Figure 3D was new. It was poly-U. The letter U stands for a nucleoside uridine. Poly-U, or polyuridylic acid, is a synthetic string of uridines, and it was used as a model compound of single-stranded RNA. According to the laureates, this compound was identified as activating the toll-like receptor 7 by another research group. And this compound was also shown to stimulate different types of dendritic cells to release inflammatory cytokines in the laureates' work published 1 year before the Milestone Article 1. I learned these from the 6th paragraph of the discussion section of the Milestone Article 1. So it was very reasonable to use poly-U as a positive control in Figure 3D. In the same figure, the primary dendritic cells were already shown to respond to another positive control, R848, an activator of toll-like receptors 7 and 8. But Figure 3D shows the poly-U did not stimulate cytokine release at all. We can see the result in the second column or the second subpanel. The reason why the cells did not respond to poly-U was not clear. The result was not described in the results section, and poly-U was discussed only briefly in one paragraph of the discussion section. It could have been due to differences in animal species used in the experiments, but again, the reason was unclear. In the professor's viewpoint later today, we will discuss my idea about why the laureates did not emphasize poly-U in the paper. Review of 4 positive controls. Can we use the term positive control? Let's discuss the 4 positive controls together. Let's view them from a different perspective in a different order. there was one beautiful positive control that worked the best. It was the unmodified in vitro transcribed RNA. It stimulated the cytokine release in figures 1 through 3 that we examined. In fact, if you look at all the figures in the Milestone Article 1, you will find that it served as a positive control in almost all figures. That is, in figures 1 through 5 and one supplementary figure. In the only remaining figure 6, it was not used. The other 3 positive controls were synthetic compounds. They were R848 that activates Toll-like receptors 7 and 8, poly-IC that activates Toll-like receptor 3, and poly-U that was thought to activate Toll-like receptor 7 and that stimulated dendritic cells of a type different from the primary dendritic cells used in figure 3D. The behaviors of these 3 were interesting. First, the behavior of R848. Only this positive control, R848, reliably stimulated the cytokine release among the 3 chemicals. In figure 2b, it was identified as a positive control for HEK293 cells expressing Toll-like receptor 7 or Toll-like receptor 8. In figure 3d, it stimulated the primary dendritic cells. So R848 was a good positive control in both figures 2b and 3d. Second, the behavior of poly-IC. In figure 2b, poly-IC was identified as a positive control for stimulating the cells expressing Toll-like receptor 3. But in figure 3d, it did not stimulate the primary dendritic cells. It was acceptable that poly-IC did not stimulate in figure 3d. This was because in figure 3d, poly-IC can be thought of as an indicator of whether the Toll-like receptor 3 was expressed in the new type of cells used in this paper. And poly-IC did not induce a positive response from those cells. Toll-like receptor 3 was not expressed at a detectable level. So to be exact, poly-IC was a positive control in figure 2b, but not in figure 3d. Third, the behavior of poly-U. In figure 3d, poly-U did not stimulate the cytokine release from the primary dendritic cells. The lack of stimulation by poly-U was a surprise, at least for me. This was because this positive control worked well in a report by another research group and in the laureates' earlier study based on another type of dendritic cells. So poly-U had been thought to be a positive control, but when tested in figure 3d, it did not work as a reliable positive control. So the compounds or treatments that are thought to serve as positive controls at some point in a paper could serve different roles in the same paper. After reviewing the behaviors of these 3 synthetic compounds, can we always use the term positive controls for the same compounds or treatments everywhere in the paper? If we do so, the readers would be confused. So I would say no. It will not be helpful for readers if the authors use the term positive controls dogmatically for the same compounds or treatments everywhere in the paper. I'll give you 2 perspectives here. 1. From the perspective of experimenters and paper readers, it will be good to think that positive controls could behave in variable manners between different papers and even at different places in a single paper. With this flexibility in thinking, we will feel more confident about designing positive controls in the experiments and discussing positive controls in the results. I think we will be able to stay flexible easily if we modify the definition of positive controls. 2 episodes ago, in episode 28, we saw that a typical definition could say, for example, a positive control is a sample where the expected outcome is known to be present. But we modified it to, a positive control is a sample where the expected outcome is thought to be present. I hope that this modified definition will give us more flexibility and more freedom so that we will manage positive controls more confidently. 2. From the perspective of paper authors, it will be good to avoid the reader's confusion. From this perspective, I would say that there are opportunities when we want to describe the experimental designs and results by using the term a positive control. It will be reader-friendly if we can write whether a specific positive control applies to a specific experiment in the paper or to all experiments in the paper. This will be a very careful approach if there is enough space for describing the details like this. There is another approach on the other end. I would like to point out that the laureates never used the terms positive controls or negative controls in the Milestone Article 1. This will avoid the reader's confusion. We are sure that the laureates were wise not to use the terms in a dogmatic manner. Professor's viewpoint. As I mentioned earlier today, PolyU was not discussed fully in the paper. Let's think about a potential reason why. Here's my personal opinion. I think that PolyU was used only as a small addition after the main experiments were conducted and completed. For example, it could happen in the following hypothetical scenario. In the original experiments, the laureates did not include PolyU as a positive control. They finished the experiments without PolyU, they wrote up a manuscript, and they submitted it to a journal. Then the peer reviewers requested the authors to include PolyU as a positive control. As requested, the laureates did a simple experiment. For example, the experiment could have used PolyU together with R848 as positive controls and lipofectin, the transfecting reagent, as a negative control. This additional experiment was performed only for the second column or subpanel of figure 3D, that is, with a specific combination of primary dendritic cells, DC2, and the released cytokine, tumor necrosis factor alpha. The laureates found that the cells did not release cytokines in response to PolyU. This new result was added to the second column of figure 3D of the revised manuscript. The revised manuscript was submitted to the same journal, and the manuscript was accepted for publication in the form that we can see now. How did I obtain this opinion, and why did I think about the hypothetical situation? That was because of some facts in the paper. I will tell you 5 facts that I found. The first fact is that PolyU was used only in figure 3D among the 6 main figures and 1 supplementary figure. So PolyU was not used systematically throughout the work. The second fact is in support of the first fact. Among the 4 panels A through D in figure 3, PolyU was used only in 1 panel, figure 3D. The third fact is that in figure 3D, PolyU was listed at a strange place among the 12 category labels that represent 12 experimental conditions. Let me explain the 12 category labels from the top. The negative control, the transfection reagent lipofectin, was listed as the first label at the top. The positive controls poly-IC and R848 were listed as the second and third labels. Below these 3 labels for controls, there are 8 category labels related to RNA 1571. And then, below all these 11 labels, there is PolyU at the very bottom. So PolyU was listed separately from the negative control, lipofectin, and 2 positive controls, poly-IC and R848. The fourth fact is, among the 3 columns or subpanels of figure 3D, PolyU was used only in the second column. Only the second column shows a horizontal bar representing the amount of released cytokines. In the first and the third columns, there are no horizontal bars. Instead, there are notes ND for not determined. So no experiments were conducted under these conditions. And the fifth fact is that PolyU was mentioned briefly in only 1 place in the text, that is, the 6th paragraph of the discussion section. Anyway, except for these facts, what I mentioned as my opinion and hypothetical scenario are just my guesses. Please note that I simply wanted to show you my thoughts about what could have happened behind the scenes. The example situation can happen to any researcher. So I thought that such a viewpoint can help you enjoy reading papers and understand some potential stories behind the presented results. Please understand that I'm not trying to criticize the paper. It is a beautifully written paper. A very good thing about Figure 3D is that the message of the paper will remain the same. It will not be affected by whether poly-U was included as a positive control or not, or by whether poly-U stimulated the cytokine release or not. Again, it is a beautifully written paper based on well-designed experiments. Now, let's wrap up. Summary. Today, we talked about the 4 positive controls in Figure 3D of our Milestone Article 1. 3 of them were poly-IC, R848, and the unmodified in vitro transcribed RNA 1571. Using these positive controls as guides, the laureates examined the release of different types of cytokines from different types of primary dendritic cells. The release of all tested cytokines from all the tested cells was suppressed when the unmodified nucleosides in the RNAs were replaced with the modified nucleosides. This was great news because it paved a way for developing messenger RNA vaccines with minimal or no inflammatory responses. We also discussed how to interpret different behaviors of positive controls. We realized that we would understand the positive controls confidently and flexibly when we modify the definition of positive controls. This is it for today. Your action. If you like this podcast, please share it with your friends, colleagues, mentors, or anybody who would be interested in it. This way, you and I can extend the reach. The bigger the audience becomes, the more feedback we can get, the better the episode quality will grow, and the more you and I can learn. Thank you for joining me in today's episode of Foundational Skills in Life Sciences. I'm Dr. Synaptologica. I'll see you soon. Bye for now.